Local currents. Local currents are the movement of ions along the neuron. An action potential occurs. As you know, an action potential is when sodium ions are entering the cell so the inside is less negative than the outside. In order for this to happen, the sodium ion channels must open and only a particular one opens at a point along the neuron. Sodium ions enter the cell. As they enter, they break the balance of ionic concentrations, as the inside of the cell has a higher concentration than the rest of the cell. Because of this, the sodium ions drift away to a region of lower concentration in the cell. They move across the cell. This triggers the sodium voltage ion channels to open, which was originally closed. More sodium ions enter the cell, so this allows further action potential to be carried along the neuron. Saltatory conduction. Saltatory conduction is basically jumping or skipping conduction. Is when the action potential jumps from one node of MVA to another. Myelin sheath is an insulating layer of fat, so it doesn't allow sodium or potassium ions to diffuse through. However, there are gaps between the myelin sheaths, which are called the node of MVA. Ionic exchanges only happen at the node of MVA because they can only enter through those gaps. So this means when the sodium ions diffuse sideways along the neuron to get to a region lower concentration, they have to move along all the way to the next node of MVA. So the action potential is jumping from one node of MVA to another, which ultimately speeds up the transmission of action potentials.